Friends, today we'll study about Brucella. We'll study about morphology, structure, and then culture, biochemical reaction, resistance, antigenic structure, biotypes, phase typing, classification, pathogenesis. Next, classification, phase typing, epidemiology, laboratory diagnosis, treatment, and prophylaxis we'll study in this video. So, Brucella. Let's begin with Brucella. The genus Brucella consists of small non-motile aerobic gram-negative cocobacilli which are strict parasite of animals, goats, sheep, cattle, pigs, buffaloes. Man acquires infection by contact with infected animals or through their products. The human disease has been recognized from very early times in the Mediterranean countries and had been known under various names such as Mediterranean fever, Malta fever, and undulant fever. Bruce in 1887, Bruce is a scientist. In 1887, isolated a small microorganism from the spleen of fatal cases in Malta. Brucella after Bruce, Malitensis after Melita, and Roman name for Malta. Bang, another scientist, in 1897 described Brucella abortus, Br abortus, in Copenhagen. The cause of contagious abortion in cattle, and then another scientist, Traum, in 1914 isolated Br Swiss from pigs in USA. These three are the major species in the genus. Three additional species, Br neotomi, Br ovis, and Br canis, have been recognized, which are animal pathogens, and except for Br canis, they do not infect man. Now let's study about the morphology. Brucella are gram-negative non-motile cocobacilli or short rods measuring 0.5 to 0.7 micrometer into 0.6 to 1.5 micrometer. They are arranged singly or in short chains. They are non-sporing and non-capsulated. Now let's study about the culture. Brucella are strict aerobes. Br abortus is capnophilic, requiring 5 to 10% carbon dioxide for its growth, whereas Br Swiss growth is unaffected by carbon dioxide. The optimum temperature for growth is 37 degree centigrade and optimum pH is 6.6 to 7.4. They may grow on ordinary media, though growth is slow and scanty. Growth is improved by addition of glucose serum, blood, or liver extract. The media employed for culture are serum dextrose agar, serum potato infusion agar, trypticase soya agar, or tryptose agar. The addition of bacitracin, cyclohexamide, polymyxin B, or vancomycin makes these media selective. On solid media, colonies are small, moist, and translucent. In liquid media, growth is uniform. Now, let's study about the cultivation in presence of dyes. The three important Brucella, Br melitensis and Br Swiss, are differentiated on the basis of their ability to grow in media containing aniline dyes, such as basic fuchsin 1:50,000 and thionin, that is 1:25,000. Then Br melitensis is not inhibited by any of these dyes, while Br abortus is inhibited by thionin and not by basic fuchsin. Br Swiss is inhibited by basic fuchsin and not by thionin. Now let's study about biochemical reactions. No carbohydrates are ordinarily fermented, but they possess oxidative capacity. They are catalase and oxidase positive. Except for Br neoteny and Br ovis, which are negative, they are urease positive, nitrate reduction positive, citrate negative, indole is not produced, and MR and VP test are negative. Urease test is very rapidly positive, particularly in Br Swiss, which is positive within 30 minutes, and Br abortus takes 1 to 2 hours to become urease positive. Now let's study about the resistance. Brucella are killed by heat at 60 degrees centigrade in 10 minutes. Hence, in milk they are rapidly destroyed by pasteurization. They are killed by 1% phenol in 5 minutes. They may survive in soil and manure for several weeks. The organism survives for 10 days in refrigerated milk, for months in butter, 1 month in ice cream. They die out in butter and cheese undergoing lactic acid fermentation. They are sensitive to direct sunlight and acid. Br melitensis may survive for 6 weeks in dust and 10 weeks in water. They are resistant to penicillin but are susceptible to streptomycin, tetracycline, chloramphenicol, and ampicillin. Now let's study about antigenic structure. Two main somatic antigens are of Brucella, that is A and M, are present in different amounts in the three major species. Antigen A is dominant about 20 times as much as M antigen in Br abortus, and antigen M is dominant in Br melitensis about 20 times of A antigen. Br Swiss has an intermediate antigenic pattern. Abortion of the minor antigenic components of antiserum will leave most of the major antibody components. Such absorbed A and M monospecific sera are useful for identification by agglutination test. The antigenic structure of Brucella is more complex. Some strains behave biochemically as Br abortus and serologically as Br melitensis, and vice versa. The battery of other tests is in addition to biotyping and serotyping is required for differentiation of various species. Antigenic cross reactions exist between Brucella and Escherichia coli, E. coli, that is 0 is to 116 and 0 is to 157. Salmonella serotype N, that is 0 is to 30 antigen. V. cholerae, PS, Maltophilia, F. tularensis, and Y. enterocolitica. Now let's study about biotypes. Br abortus 7 biotypes, that is 1 to 9 number, 7 and 8 are deleted. Br melitensis, that is 3 biotypes. Br Swiss 5 biotypes. Br Swiss strains which produces H2S are called American strains, and those not producing H2S are called Danish strains. Now let's study about the phase typing. The Tbilisi, in short TB, reference phase is used for phase typing. It lyses abortus at both RTD routine test dilution and 10,000 RTD, but Br Swiss is lysed at 10,000 RTD only, while Br melitensis is not lysed at all. Now let's study about the classification. Brucella may be classified into 6 species based on carbon dioxide requirement. Production of H2S, sensitivity to aniline dyes, basic fuchsin and thionein, agglutination by monospecific sera, and phase lysis. Now let's study about pathogenesis. All the three major species of Brucella are pathogenic to human beings. BR melitensis is the most pathogenic species, followed by BR suis in intermediate pathogenicity, and BR abortus is the least pathogenic. Brucellosis is a zoonotic disease. Next is, let's know about the mode of infection. Human infection occurs by direct or indirect contact with infected animals. Now let's study about this mode of infection in details. Brucelli are transmitted to humans by drinking contaminated raw milk or by ingestion of milk products from infected animals. Direct contact with infected animal tissues, butchers, dairy workers, farm workers, and veterinarians are particularly at risk. Accidental ingestion, inhalation, injection, and mucosal or skin contamination may occur in laboratory personnel working with Brucelli. Now let's study about types of infection. Human infection may be of three types. First is subclinical or latent infection. That means there is no clinical evidence of disease but is detectable only by serological test. Next is acute brucellosis. It is also known as undulant fever or Malta fever. It is associated with prolonged bacteremia. Now let's study about the chronic brucellosis. It is usually non-bacteremic. The symptoms are generally related to a state of hypersensitivity. Now let's study about the course of disease. The bacilli, after entering the body, pass via the lymphatic channels, regional lymph nodes, and thoracic duct to the bloodstream, causing bacteremia. Brucellosis is primarily a disease of reticuloendothelial system, hence the organisms are localized in the RE system. Brucella have a special predilection for intracellular growth, especially inside phagocytic cells. Cell-mediated immunity plays an important role in recovery from brucellosis. The incubation period varies from 2 to 3 weeks and may extend to 6 months. The onset of symptoms may be acute or insidious. Acute brucellosis is characterized by fever, chills, severe malaise, severe headache, bone and joint pain, mild lymph node enlargement, and sometimes hepatosplenomegaly. The name undulant fever has been attributed to acute brucellosis because of the periodic nocturnal fever that may occur over weeks, months, or years, especially in untreated cases. When the disease persists for 6 months or more, it is termed as chronic brucellosis. Now let's study about the epidemiology. The disease is worldwide in distribution and endemic in certain areas such as Mediterranean countries. Certain swine rearing areas of USA and Greater Britain. In India, BR melitensis is the predominant pathogen of human brucellosis acquired from goats and sheep, which is followed by BR abortus of cattle origin. Now, let's study about the laboratory diagnosis. Brucellosis is a febrile disease with varied clinical manifestations, and clinical diagnosis is often difficult and laboratory aid is therefore essential. Laboratory methods include culture of Brucellosis, serology, and hypersensitivity test. Now let's study about the specimens. Blood culture is the most definitive method for the diagnosis of acute brucellosis. Brucella can also be isolated from bone marrow, liver, lymph nodes, and occasionally from CSF, urine, sputum, breast milk, vaginal discharge, and seminal fluid. Now let's study about blood culture. Blood is collected during the pyrexial phase because chances of positive blood culture are more during this period. 5 to 10 ml of blood is inoculated into 50 to 100 ml of serum dextrose broth or trypticase soya broth and incubated at 37 degrees centigrade aerobically in presence of 5 to 10% carbon dioxide. Subcultures are made on solid media every 3 to 5 days for 8 weeks and incubated at 37 degrees centigrade aerobically in presence of 5 to 10% carbon dioxide. The need for frequent subcultures can be avoided by use of Castaneda's method of blood culture. It contains both liquid and solid media in the same bottle. The blood is inoculated into the broth and the bottle incubated in the upright position. For subculture, the bottle is titled so that the broth flows over the surface of solid agar slant. It is again incubated in the upright position. In case of positive blood culture, colonies appear on the agar slant. This method reduces the chance of contamination and risk of infection to laboratory workers. Blood cultures are positive in 30 to 50% cases even when repeated samples are tested. BR melitensis and BR suis are readily cultured than BR abortus. Bactec BSc-TEC is a rapid method for culture and becomes positive within 5 to 6 days. Now let's study about the identification. It depends on biochemical test, dye test, agglutination with monospecific sera and lysis by phage. Now let's study about the serology. Serological methods are more useful as cultures are negative in high percentage of cases. Antibodies, that is IgG and IgM, appear in 7 to 10 days after clinical infection. As the disease progresses, IgM antibodies decline while the IgG antibodies persist or increase. The chronic brucellosis, only IgG can be demonstrated and IgM may often be absent. The agglutination test identifies mainly the IgM antibody, while both IgM and IgG can fix complement. IgG and IgA antibodies may act as blocking, that means incomplete antibodies, and may prevent agglutination. The agglutination test is usually positive in acute infection but may be negative or weakly positive in chronic cases. Now let's study about the standard tube agglutination test. This is a tube agglutination test in which equal volumes of serial two-fold dilutions, that means 1:20 to 1:640 of patient's serum and the standardized antigen, that means heat killed Brucella suspension, are mixed and incubated at 37 degrees centigrade for 24 hours. A single tire of more than 1:160 is presumptive evidence of Brucella infection. Now let's study about the prozone phenomenon. Let's know about the prozone phenomenon. Prozone phenomenon is very frequent in brucellosis. Therefore, it is essential that several serum dilutions should be tested. Another source of error is presence of blocking or non-agglutinating antibodies in serum. The blocking effect may be removed by heating the serum at 55 degrees centigrade for 30 minutes or by using 4% saline as diluent for the test. The most important method for detecting these incomplete antibodies is the antiglobulin test, that is Combs test. A positive agglutination test may also be produced by cholera, tularemia, or Yersinia infection or vaccination. Cholera-induced antibodies may be removed by absorption with 2-mercaptoethanol. Agglutinin titers are expressed in international units. This is done by using a standard reference serum for comparison. Now let's study about Castaneda strip test. A loop full of patient's serum is placed on a filter paper strip containing a colored Brucella antigen. Antibody is able to fix the antigen and prevent its movement along the paper. It is simple and suitable test for screening of brucellosis cases. Now let's study about the complement fixation test. Complement fixation test. In short, CFT. It is more useful in chronic cases as it detects both IgG and IgM. Now let's study about ELISA and DIA. These tests are very sensitive and helpful to distinguish acute and chronic brucellosis. ELISA can detect IgM and IgG antibodies separately. Now let's study about the indirect immunofluorescence test. It is specific and sensitive method for detecting antibodies and may be positive even when the agglutination test is negative. Now let's study about hypersensitivity test, or we can say Brucellin test. It is displayed type 4 hypersensitivity to Brucella antigen. It is an intradermal test using protein extract of the organism called brucellin. Positive reaction is indicated by an erythema and induration of 6 mm diameter within 24 hours. This test is not useful for diagnosis of acute infections because test may remain positive for years after acquiring infection. It may be helpful in chronic brucellosis where a positive skin test may sometimes be the only indicator of infection. Persistent negative skin test helps to exclude brucellosis. Now let's study about the detection of animal infection. The methods used for the laboratory diagnosis of human infections may also be employed for the diagnosis of animal infections. Culture of milk and urine may give positive results. In addition, rapid methods such as rapid plate agglutination test and the Rose Bengal card test have been employed for the diagnosis of brucellosis in herd of cattle. For detection of infected animals in dairies, besides culture, pooled milk samples may be tested for antibodies by milk ring test. Now let's study about the milk ring test. It detects Brucella agglutinins in the milk of infected dairy cattle. Now let's study about the methods. A sample of whole milk is mixed well with a drop of stained Brucella antigen. A concentrated suspension of Br abortus or Br melitensis stained with hematoxylin in a narrow test tube. This mixture of milk and bacterial suspension is incubated in a water bath at 70 degrees centigrade for 40 to 50 minutes. Now let's know about the result. In a positive test, the bacilli are agglutinated and rise with the cream to form a blue ring at the top, leaving the milk unstained. When antibodies are absent, that means negative test, no colored ring is formed. The milk remains uniformly blue. When agglutination test is another useful test for detecting antibodies in milk. Now let's know about the treatment. Treatment consists of administration tetracycline along with streptomycin for a period of not less than 3 weeks. WHO has recommended a regimen of ripapicin 600 to 900 mg and doxycycline, that is 200 mg, both taken as a single daily dose for a minimum of 6 weeks. Now let's study about the prophylaxis. Persons handling the animals should use protective clothing and gloves. pasteurization or boiling of milk should be done. Now let's study about the vaccination. Cattle should be vaccinated with live attenuated BR abortus strain, that is 19 vaccine, and goats sheep with BR melitensis Rev 1 vaccine. Unimmunized infected animals should be slaughtered. Then next point is BR abortus strain, that is 19 to 19BA. A more attenuated variant of strain 19 has been widely employed for human immunization in USSR for protection of population exposed to infection. Vaccine is given intradermally. Finally finished. Any questions regarding this, you can ask me in the comment box, and subscribe my channel to know more updates. Thank you.